Hey everyone, and welcome to this explainer. I am seriously thrilled to have you here today because we're about to decode the incredibly complex microscopic security systems running our entire bodies. If you've ever found yourself wondering how the physical boundary of a single cell can dictate exactly what you feel, how you move, and how medicine actually heals you, well, you're in the exact right place.
Think about this for a second. How do deadly frog toxins, the addictive pull of nicotine, and life-saving anti-anxiety medications all manage to work in the exact same way? I know, it sounds like the setup to some weird biology riddle, right? But the answer is actually remarkably elegant, and it all comes down to understanding the tiny, molecular bouncers guarding the doors of our cells.
So here's our roadmap for today. First, we'll cover the cell's security gates. Number 2, chemical keys, Ligand-Gated channels. 3, hijacking the brain's receptors. 4, electric triggers, voltage-gated channels. And finally, deadly toxins and life-saving drugs.
All right, let's jump right into part 1, the cell's security gates. You know, our cells are surrounded by a strict barrier to keep the outside world out, but obviously for a cell to actually function, it needs to communicate and absorb specific elements. Enter ion channels. Think of these as highly exclusive security gates embedded right into the cell membrane. They absolutely do not just let anything through. Instead, they're perfectly designed to rapidly transport very specific charged ions. We're talking sodium, potassium, calcium, or chloride. By controlling this flow of cellular electricity, these gates literally dictate our senses, our muscle movements, and even our most complex emotions.
Just look at the sheer mechanical difference between the two main ways a cell moves stuff across its border. You've got a transporter, which has to bind to a solute, physically change its entire shape, and then drop it inside. It's effective for sure, but it's kind of slow. Now compare that to a channel protein. That thing is just a direct open gate. When that pore opens, ions flood through instantly. It's this rapid-fire action that allows you to instinctively pull your hand away from a hot stove in just a fraction of a millisecond. These fast-acting gates basically fall into two distinct categories based on how they actually unlock. First up, we've got ligand-gated channels, which are essentially physical locks that need a very specific chemical key to pop them open. And second, we have voltage-gated channels. These don't use a physical key at all. Instead, they rely on an electronic override, triggering wide open in response to changes in the cell's own electrical charge.
Which brings us to part 2, chemical keys or ligand-gated channels.
The big takeaway here is that a ligand-gated ion channel is incredibly stubborn. It simply will not open unless the absolutely perfect chemical key slides into the lock. These keys are usually small extracellular organic molecules, mostly our own neurotransmitters like acetylcholine or GABA. So the right neurotransmitter binds, and boom, the gate springs open. But timing is literally everything. There's this crucial inactivation period, where the channel just shuts down to reset. During that window, it absolutely cannot receive a new signal or ligand. It's kind of like nature's built-in spam filter, keeping the cell from getting totally overstimulated.
Let's connect this microscopic mechanism to something super macro and relatable: human addiction. Deep within our brain's dopaminergic neurons, we have these specific ligand-gated channels called alpha-4-beta-2 nicotinic acetylcholine receptors. Normally, our body's own naturally produced acetylcholine opens these up to manage our stress and reward systems. But it turns out nicotine is essentially a master skeleton key. It fits flawlessly into this exact channel, hijacking the whole gate to artificially flood the brain with reward signals. In fact, if you block this specific gate in lab mice, you completely decrease their reward response to nicotine.
But nature is pretty clever, right? Check out the laburnum flower. This little plant contains an alkaloid called cytosine, and cytosine happens to have a shape that's incredibly similar to nicotine. This means it can actually compete for that exact same microscopic keyhole. By physically sitting in the receptor, it completely blocks the nicotine from binding. It's a fantastic example of how we might break chemical addictions just by swapping out who holds the key to the cellular gate.
Moving right along to part 3, hijacking the brain's receptors.
Say hello to the GABA receptor. If the channels we were just talking about are the gas pedals of the nervous system, GABA is literally the biological brake pedal. It's a ligand-gated channel that specifically lets negatively charged chloride ions rush into the neuron. All that negative charge cools the cell down, stopping it from firing out of control. So when this specific gate gets dysfunctional, the brakes basically fail, which leads to some serious conditions like epilepsy, chronic pain, and severe insomnia.
Now, when you really look at the architecture of the GABA receptor, you'll see it's way more than just a simple keyhole. There are actually multiple, highly specific parking spots, known as allosteric binding sites, scattered around the receptor.
You've got the main spot for GABA, sure, but then there are entirely different spots where completely other molecules can dock to influence just how wide the gate opens and for exactly how long.
So what exactly is parking in those extra spots? Honestly, some incredibly familiar substances. Benzodiazepines, like diazepam, park in one spot to treat anxiety. Barbiturates park in another to act as heavy sleeping agents. Even volatile anesthetics like chloroform or everyday alcohol interact directly with this exact receptor complex. All of these wildly distinct substances are basically doing the exact same thing. They are helping force that GABA gate open wider or longer, letting more of those calming negative chloride ions into the cell, which results in profound sedation.
This is where things get wild. Look at the massive difference in chloride flow here. Normally, GABA gives you this nice, steady, regulated stream of chloride to keep you calm and balanced. But under the influence of acute alcohol, the channel basically gets stuck in overdrive. The gate just gets flooded, and that massive rush is what causes the heavy depressive effects on the central nervous system that we associate with intoxication.
But hold on, what if we go the opposite route? Imagine instead of holding the brake pedal down, you just completely cut the brake lines. That is exactly how a lot of potent pesticides work. Chemicals like fipronil, which you might actually recognize from your dog's flea medication, or TETs, a banned rodenticide, act like cellular superglue. They jam this exact GABA channel totally shut. Without any way for those negative chloride ions to get in and cool things down, the nervous system gets thrown into a state of catastrophic overload and deadly hyperexcitation.
All right, part 4, electric triggers, voltage-gated channels. Now, unlike those chemical locks we just explored, voltage-gated sodium channels, let's call them NAVs for short, do not care about chemical keys at all. They're ultra-sensitive electronic security gates. The moment they detect even a slight shift in the electrical voltage of the membrane around them, they pop right open. This lets a massive rush of positive sodium ions flood into the cell. And this specific electrical spike, known as depolarization, is the literal spark that powers every single thought, heartbeat, and physical sensation you have ever experienced.
The actual step-by-step mechanics of this? Totally mind-blowing. First up, the surrounding cellular voltage shifts. Then, deep inside the channel, a specific piece of the protein that's loaded with positive amino acids physically repels away from that changing charge. This physical movement literally yanks the gate open in step 3, letting all that sodium rush in. But step 4 is arguably the best biological hack ever. An actual protein chain, acting almost like a ball on a string, instantly swings up and plugs the pore. This inactivation ball stops the signal dead in its tracks, making sure the electrical spark is a quick, precise flash, rather than a continuous, completely damaging shock.
And finally, part 5, deadly toxins and life-saving drugs.
Because these voltage gates are literally keeping us alive, they happen to be the absolute favorite targets of nature's most brutal assassins. Tetrodotoxin, which you find in pufferfish, works by perfectly corking the sodium channel pore. It fatally inhibits the gate, meaning your electrical spark of life just can't fire at all. On the flip side, batractotoxin from poison dart frogs does the exact opposite. It binds to the channel and sensitizes it so much that it locks completely open. The cell fires once and then can never ever reset. While their methods are total opposites, both of these toxins devastatingly destroy neuron signaling, leading to rapid, fatal paralysis.
But here is the silver lining, the real triumph of modern science. We are using these exact same deadly mechanisms to actually save lives. By carefully inhibiting these sodium channels with drugs like Lidocaine, we can block the electrical signals of localized pain. Or we use lamotrigine to prevent the chaotic electrical storms of epilepsy. We can even target similar voltage-gated calcium channels with drugs like verapamil to relax smooth muscles and lower blood pressure, or use specific activators to correct irregular heartbeats. We are literally in there, manually adjusting the dials on our body's electrical grid.
Which pretty much brings us to the edge of the map. We've mapped out a lot of these cellular security gates, but honestly, there are still countless receptor combinations and structural mysteries that remain totally or at least poorly understood. Which leaves us with a pretty thrilling question. In the vast, completely unexplored microscopic frontier of our own cells, what future cures or undiscovered deadly toxins are just sitting there waiting for the right key to unlock them? Thank you so much for joining me on this explainer. Keep wondering, and I'll catch you in the next one.